So a second approach then is the recognition that there are protective niches both in the bone marrow and in the lymph node, where these are very complex cellular biological entities in the bone marrow and in a lymph node, and that the CLL cells are actually supported and promoted. Their growth and expansion is promoted by what's in there. And part of it is this close association of CLL cells with these nourishing cells. So if you take these large spheres with a nuclear indication, these are indicated as the dividing cells or the proliferating cells. They're associated with another cell type that nourishes them. These are the more indolent CLL cells that aren't growing. So then at this point then, it's known that cells in the bone marrow are protected from some of our best chemotherapies. So for instance, fludarabine is not very effective when these cells are in close alignment with these what's called stroma or microenvironment. And then those cells become less susceptible to fludarabine. And then essentially the clinical outcome of that is that the bone marrow is not cleared of leukemic cells. And the same is true with the lymph nodes. But we now know that there are molecules that can dislodge, come in between the adhesion molecules between the CLL cells and these nutrient cells, dislodge those cells and get them out into the circulation. So they're no longer directly supported by the microenvironment, and they're more susceptible to cell killing. So this is demonstrated, and this, again, is a clinical strategy that's being brought forward as well. So there are several, so these are general biological properties of chronic lymphocytic leukemia. There are some specific properties that can be seen by evaluating the chromosomes. And you've probably heard this terminology of P17, 17P minus, and 11Q minus, and 13Q minus. I'm going to deal with each one of those because they have a particular attribute that endows CLL cells with some of its pathophysiological properties. So probably the most widely recognized and most consequential is the P17P deletion. So this is the site where what's called a tumor suppressor gene, P53, is located. And it has the role not only in the context of CLL and the therapies that we're speaking of, it's effective in allowing those therapies to work. And in the absence of this protein, P53, then those therapies don't kill the cells effectively. So I thought it would be interesting to see what the hematopathologists see, in that this is a microscopic slide of chronic lymphocytic leukemia cells. And what you may be able to see from the back, I could see it great when I was sitting at my desk with a screen in front of me. You can see it here. That some of these cells have two of these pink dots. So the probe for that 17P region, which is deleted in some of these individuals, is present. So we have one chromosome from a mom and one from our dad. So 2 is the normal situation. But in this situation, this patient also had, this cell scan, also have cells that show a deletion in P17. Now, the thing that's become recognized in the last year or so is that the remaining allele, say if one is deleted and then there's one remaining, you could say, well, why doesn't that work? Well, the reason it doesn't work has been published and recognized is that it's mutated and inactive. So almost all of the individuals who show the 17P deletion have an inactive P53 function, and it's critical to respond to therapy. And here's something that was published this week from the German cooperative groups showing in an earlier study of fludarabine and Cytoxan. This is the survival time, fraction alive, of patients with normal P53 function, and then this is with patients with a mutation in P53. I think that's pretty dramatic. I've not seen something like that before. But so this is something that's coming out of these large randomized trials that were generated by the work that was done at MD Anderson originally. So what do we do about it? We're supposed to be talking about treatments here. So just briefly, I'd say that there is another gene of the same family as P53, but it's not expressed very well in CLL. It's called P73. But we found a way to wake that up and get it to be expressed. And then what the working hypothesis is that if you have P73 expressed in CLL, then it will be able to respond to cytotoxic drugs such as fludarabine and Cytoxan. And that's being pursued actively now as well. So resensitizing cells. The second chromosomal deletion is this 11q. Leads to a loss in DNA repair capacity. Is a different depiction of a chromosome. There's a short arm and a long arm, and this is where this deletion takes place. So the Q arm is the long arm, the P arm is the short arm, and this contains a gene called ATM, which is essential for DNA repair capacity. Now, importantly, Dr. Keating and his colleagues have found that more than half of the patients with CLL who fail to respond to, fail to maintain a response to FCR therapy exhibit this deletion. So that gene is gone. And we know now also that by sequencing the residual allele, that protein is inactive and largely mutated. More than half of those patients are. So this is a problem, so then we were thinking, well, is there anything that we can do where that particular function is needed or the cells die? So that is, you look at DNA repair capacity as a resistance mechanism for your drugs. So now we're switching it around a little bit. What it shows is a survival curve that was done in cells that were done in the laboratory, but it shows between a 50 and 100-fold sensitization in cells that lack this protein ATM. They're much more sensitive than cells that do have the ATM gene. So what it means is that there is specificity of the treatment conferred against the tumor by 50 to 100-fold. And Bill Weirder will be the PI of this protocol that we're bringing forward and hoping to evaluate this. The last deletion, chromosomal indicator is this 13q deletion. And this is the most prevalent of the deletions that's found in CLL. And it's, one of our colleagues has recognized and found that there is a regulator of these pro-survival proteins that's present at this site. And those regulators are deleted in patients who have this particular deletion, and that explains why CLL, why these pro-survival proteins are so prevalent in CLL. And he's also bringing forward a rationale to address this by they're called microRNAs, and he has a rationale that he's brought forward in mice to show that if you use this particular type of restoration therapy, then the pro-survival proteins decrease and the disease diminishes. So these are some of the strategies for targeted therapy. I hope I've tried to explain the target in biological terms, and it's not just an individual, but it largely could be a group, likely will be a group of individuals who's targeted with that. Thank you very much.